electrophoresis is a method used to separate molecules based on their size. The objective of this experiment is to identify and quantify the molecular weight and concentration of unknown proteins by running them in an SDS page gel. A typical polyacrylamide gel has two main parts, the resolving gel and the stacking gel. The resolving gel is the bulk of the gel where the separation occurs, while the stacking gel is a layer on top with wells in which protein is loaded. The gel is placed in a chamber that is partially filled with buffer solution, and a negative electrode is attached to the top of the gel, while the positive electrode is attached to the bottom. A voltage is then applied, allowing the protein to travel down the gel towards the electrode of the opposite charge based on the size of the protein. To eliminate the effects of protein structure and charge, SDS detergent is used to denature the proteins and give them uniform charge to mass ratio. Larger proteins travel slower, causing them to stay toward the top of the gel, while smaller proteins are able to travel further. After the proteins are run, the gel can be imaged and analyzed using the software Image Lab to determine the protein molecular weight, identify unknown proteins, calculate protein concentration, and determine protein purity. Before making the gels, the glass cassette apparatus is cleaned and assembled. The resolving solution is made by mixing 3 milliliters of Resolver A, 3 milliliters of Resolver B, and 30 microliters of 10% APS, and 3 microliters of Temid. The solution is added between the glass plates, overlaid with isopropanol, and then left to polymerize for about 5 minutes. While this is happening, the stacking solution is made by mixing 1 ml of Stacker A, 1 ml of Stacker B, 10 microliters of APS, and 2 microliters of Temid. The resolver gel is dried, and the stacking gel is pipetted over the resolver gel. The comb is inserted into the gel and is left to polymerize for about 30 minutes, or until solid. 3 protein samples, protein K, A, and J, are removed from the freezer and thawed. For each protein, 100 microliters of protein, 50 microliters of lamellae buffer, and 50 microliters of beta-mercaptoethanol are added to a microcentrifuge tube. The beta-mercaptoethanol was added and capped as quickly as possible due to the strong, unpleasant odor. The samples are then heated in a dry bath for 5 minutes at 95 degrees C. The comb is removed and proteins are loaded into the gel with the volumes in order shown. Once the proteins are loaded, the electrophoresis cell is assembled and the buffer solution is added. Before turning on the voltage, it was ensured that no contact was made with the chamber or wires, as shocks could occur. The cell is set to 300 volts and the gels are run until the solvent front reaches the bottom. The gels are carefully removed and then analyzed. First, gels 1 and 2 were run. When gel 1 was imaged using Image Lab, it was clear that the proteins were loaded at too high of a concentration because they appeared red, indicating that the image was oversaturated. To resolve this, the proteins were run in lesser quantities in the later gels. In this image, you can see a large amount of J remained in the top well. This indicates that the molecular weight of J was too large and ran out the range of the gel. Proteins A and K were then run alongside the standard. The lanes and bands tool in Image Lab was used, and then the standard lanes were specified under the molecular weight tool. The molecular weight tool then provided the kilodalton values for the standard, allowing us to determine the molecular weight of unknown proteins A and K. A and K had the same bands, differing only in their intensity because of their varying protein concentration. The bands align around 27 kilodaltons and 30 kilodaltons, translating to 27,000 grams per mole and 30,000 grams per mole. Gel 3 was loaded with different concentrations of protein K. In lanes 1, 3, and 5, the dilutions are 35, 25, and 15 microliters respectively. The intensity bands were found using the program, and the width of the tallest peak was adjusted to fit the curve. Using the volume intensity of the largest band for each dilution, a calibration curve was constructed plotting volume versus protein mass, and a trend line was fit. The concentration of protein A can be determined using the equation of the trend line. In the equation, the volume of intensity A can be solved in for Y, allowing you to solve for X, the protein mass. The protein mass can be divided by the volume of protein used in that dilution, giving the protein concentration. This was done for each of 2 dilutions of A, and the average concentration was calculated to be 0.32 milligrams per milliliter, compared to K, which had a concentration of 0.25 milligrams per milliliter. The protein purity was calculated by taking the volume intensity of the 2 protein bands and dividing it by the total volume intensity in that lane. For protein K, the average protein purity was found to be 25%, while protein A was found to be 51% on average. We determined that proteins A and K were both casein, a common milk protein. The protein bands seen in our gel match with the proteins in this gel found in literature in both appearance and molecular weight. Protein A was found to have a concentration of approximately 0.32 milligrams per milliliter, which makes sense because in our first gels, A appeared oversaturated when loaded in the same quantity as K, indicating that A had a higher concentration than K. Protein K was 25% pure, while protein A was 51% pure. Protein J had too high a molecular weight to run in our gel, so we were unable to identify it. Due to the fact that we ran effectively one trial, the data is not statistically valid. Further trials could be run to increase the soundness of the data. During imaging, there was a considerable amount of error involved in determining the band intensities, increasing the overall uncertainty in numerical results.